Dr. Josep Samitier.
Very nice to meet you.
Here you go.
Thank you. Thank you very much to the organizers for this option to speak a little about the activity in the Institute for Bioengineering of Catalonia. So the title of my presentation is 3D bioprinting, the challenge and the possibilities, and a little what is our experience in the field. So the first question is, what is bioprinting? Bioprinting is the process of generating a controlled cell pattern in 3D, combining materials and cells all together. So this is in the picture. We can observe the use of soft materials together with cells and try to build a 3D structure combining different materials and also different cells. This is after the bioprinting process. It's possible to observe the structure and using a microscope by fluorescence, observe where are located the cells. But it is more or less an academic exercise. The question is how to try to mimic our organs, because in the human body, it's clear that the humans are 3D. So the interaction between the cells are not in 2 dimensions, it's in 3 dimensions. 3D cultures show a more realistic cell biology and function, and many of the problems between when we move from the usual 2D cell cultures to the animal models, and finally in the humans, many of the problems observed is because the 2D cell cultures are not fit very well all the characteristics of the physiological body. And obviously, the obtention of 3D biological models will provide more reliable data, accelerate the co-synthesis process, and also reduce the animal experiments. So during many years, scientists tried to develop 3D cell cultures using different technologies, for instance, explants, using transwell, spheroids, and also the usual 3D printing of scaffolds, and after try to introduce the cells inside of the scaffolds. The main problems and drawbacks of these technologies, the limited control of the cell distribution, and also one of the main problems at this moment is the vascularization. So how introduce the porosity and the capability of the blood vessels inside of this 3D cell tissue. So in principle, the idea of 3D bioprinting is very similar to 3D printing. So more or less, it's try to obtain a mixer introducing the material and the cells, and to use a robotically controlled pastry bag, and that's more or less similar to the all 3D process. But the reality is that it is a bit more challenging because you need to use multi-material printing, because the different tissues and organs contain different extracellular matrix with different properties, with different stiffness, with different porosity. Also, we need to use soft materials because it's not compatible with the cells, and try to build 3D extractors using very soft materials is a challenge. And finally, how to combine the stimulus for the cells and how to obtain a system that can be mimetic or biomimetic for the cells, and this system can evolve in the time. So it's clear that to perform a good 3D bioprinting process, you need to develop first a 3D model, to use the technological bioprinting system, it's necessary to work in sterile conditions. The bioink is essential. How is the right combination of different biomaterials, artificial or natural biomaterials, which can be combined with the cells? What are the cells used? And many times, to try to develop a tissue, you need to combine different types of cells in the co-culture process. And what is the time to develop the printing, to perform the maturation of the structure. And this is very relevant, the combination of the 3D bioprinting together with the bioincubators. So Here is an example of the different options for microextrusion bioprinting using piston, pressurized air, screw, and it is possible to produce this combination of biomaterials and cells. But in the recent years, different scientific publications has analyzed different reviews about the different type of bioinks, biomaterials that can be used, synthetic or natural. They use also to the combination of structural biomaterials together with sacrificial biomaterials in order to produce and to help in the process of the build of the 3D extractor, and also what will be, for instance, the support bioink. So it's not only one type of materials that can be used, you need to combine different biomaterials in this complex 3D engineering process. So first of all, you need to develop the model, to develop a 3D CAD model that can be after to import to deal with complex shape, and finally to go to the 3D printing in order to produce the structure. Many times using first the decom format from the medical needs. And these are an example of a review of different structures produced and published in the literature concerning using different types of materials, chitosan, alginates, agarose, fibrin, gelatin, and more complex type of hydrogel, depending on that. So last year, I had the opportunity to participate in the first edition of this industry fair. And in this case, I explained some of the last results, for instance, from the WIS Institute, using, in this case, 3D bioprinting with multi-cartridge models in order to combine all of these materials in a 3D construct, combining structural materials with sacrificial materials. And using this approach, will be possible to produce, for instance, printed air construct, in this case using chondrocytes in this process, and it is possible to observe where the option to produce the microchannels for porosity, to introduce different type of cells, and also introduce also a structural material in order to help in the construction of the system. And finally, we obtain a chondrocyte-laden hydrogel with the possibility to mimic the air of cartilage. Using the same process, it's possible to produce bone, in this case for a bone defect, and use, in this case, pluronic as a sacrificial material, and that's the same process, or also to produce bioprinting muscle using, in this case, 3D extractors. So this is a little the state of the art. And what is our experience in IBEC? So IBEC is an institute for bioengineering. So we work with biological material, and our focus, one of the main axes, is tissue engineering. So all the problems concerning 3D cell cultures, development of scaffolds, and combination of biomaterials with cells and stem cells. In this sense, also, as engineering part, we use many different types of technologies. One of them, we are specialized in 3D bioprinting. So you have at this moment in our facilities different type of bioprintings. The main equipment is this equipment from Regenou, but we have also other small bioprintings in the framework of a biospace, because we need to combine the printing process with the culture process in order to produce finally the 3D tissue. So the system that you use is a macroextrusion system using inkjet printing, and it is possible to cool and heat the different bioinks in order to stabilize the temperature, in some cases at 37 degrees. So IBEC at this moment are more than 300 people, 20 labs, and in red are all the labs in our institute that at this moment are using 3D printing or 3D bioprinting in the activity. So more than half of our institute is focused at this moment in the development and the use of 3D printing or bioprinting process for health purpose. So we are also members of the, and participate in the initiative from the Catalan government about the 3D printing hub that will be located here in Barcelona, and in the framework, obviously, of 3D bioprinting. And I will try in the next slide to show some of the contributions that at this moment we are working. So, for instance, one of the first questions in 3D bioprinting is try to develop the cells, and in this case, the IPS, and the evolution of the IPS and the differentiation of IPS is essential. For instance, in the case of cardiac tissue, you try to develop a 3D cardiac tissue. One option is use first human embryonic stem cells in order to produce the cardiac differentiation. In this case, Dr. Nuria Montserrat is our team leader in our institute to produce that. After, you need also to develop 3D bioreactors that we will introduce when you produce the 3D printing tissue inside the bioreactors in order to stimulate the culture in order to produce the differentiation of the tissue. And using this system, it is possible. This is the control of the 3D tissue obtained after the differentiation of cardiac IPS. But if you work right, you arrive to obtain this type of system after the electrostimulation inside of the bioincubator. So it is possible to observe the strong contraction of the maturity of the 3D patch, in this case, of the system. But the more relevant is that if you analyze the electrocardiogram obtained from the 3D patch, this electrocardiogram is very similar to the cardiac electrocardiogram of the organ, with the difference between the control where the cells are not well stimulated, they are not well interacting between them, and in this case, the stimulus are very bad. So, using the right weight of 3D printing together bioreactor together with the cells, it is possible to mimic and to produce in vitro a patch that has the same answer that a 3D cardiac tissue. So in this case, it is possible to analyze in vitro, for instance, for purpose of drug development, to induce arrhythmia inside of this 3D patch and analyze the behavior of this arrhythmia and using different type of drugs. So this is an example of the possible application of this patch. Obviously, the idea is after try to use this cardiac patch for chirurgical purpose. So that is the next step. So this is a little what is the objective of, in this case, of the use of this combination of IPS, 3D scaffold using by bioprinting, the culture of the bioreactors, and finally the obtention of the microscopic cardiac tissue construct. We can move, it is possible to produce cardiac tissue, it's also possible to produce skeletal muscle tissue. And in this case, these are the hydrogels obtained by 3D bioprinting, in this case without the cells, but it's possible to combine that in a 3D, and in this case, you can obtain the myotubes, so muscle fibers that can be used after for 3D purpose. So, another application is in the case of the osteoarthritis, so the problem of degeneration of the cartilage. In this case, this is a problem because the cartilage don't regenerate, and it is difficult to obtain a good regenerative therapy. In our case, we have developed a system that induces the differentiation and the chondrogenesis of the mesenchymal stem cells, and we have using also this 3D structures, induce that inside on an animal model, and it is possible that in this case that is a combination of 3D bioprinting together with nanotechnology arrive to obtain a good regeneration of the cartilage in the tissue. Another example, so you are working at this moment in the Advanced CAT, an initiative of the communities of USTRY in Catalonia, and this community in health has a project concerning advanced therapy, and inside of that we are working in the development of 3D bioprinting for tendon, and in this case you use different types of mesenchymal stem cells and tenocytes in order to induce 3D regeneration of tendon. Finally, the last example that we try to explain is one very interesting, exciting approach that the use of the 3D bioprinting combining natural bioinks, and in this case using the cellularization of the organs. This is a work recently published in our institute with Dr. Núria Montserrat, together with the collaboration of clinicians and Dr. Josep Maria Campistol from the Clinic Hospital, or Dr. Francisco Fernández Avilés from Madrid. And in the framework of this work, you have worked in the development of 3D patch for kidney tissues. So the first option is try to develop the cellularization technology of the natural organ in order to use that as a basis for the bioink material. After that, try to develop as a model organoids, and this can be possible to develop in last year, in 2016, kidney organoids, which reproduce the structure of kidney. The next step has been to analyze and to induce the models of kidney inside a 3D bioprinting system. And finally, the main objective of the project is to combine patient-specific IPS together with renal progenitors, patient-derived kidney organoids, use the cellularized kidney extracellular matrix from biomimetic hydrogels, use the bioprinting process, and finally obtain this human kidney graft that can be used after for the surgeon for different type of kidney disease. So obviously there are many challenges to develop that, and has been explained before my talk. Obviously that will be the process at this moment in the lab, but probably in the next 10, 15 years that will be available in many of the hospitals worldwide. Some of the specific challenges that you need to address in each of applications is the development of cell-compatible hydrogels with the right properties, viscosity, biochemical composition, the monitoring of the bioink flow rate, the alignment, the automatization of the system. So there are biological problems together with the engineering process. So the collaboration with the people who develop new systems, new devices, new equipment is extremely relevant, together with the people that combine the materials and the cells in the process. And the development of bioreactors for tissue organ ground and maturation after the first deposition. Basically, it's clear that the future outcomes will be the increase of the printing fidelity, the rapid production of the 3D printed constructs, and the generation of functional tissue organs that can be used by the surgeon. So thank you very much for your attention.
Very good.